Welcome to the video on body fluids. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. Let's start with a thought-provoking question. Which body fluid do you produce the most of in your lifetime? Here's a riddle. I can sip my blood, wash away dust, or spill when emotions combust. What am I? Think about this as we move forward. Our learning objectives cover several key concepts. We will describe the concept of pH, the Henderson-Hasselbalch equation, and the role of various buffers in controlling body pH. We will also describe the distribution of body fluids, differentiating between intracellular and extracellular fluids, and define surface tension, viscosity, and osmosis, along with their physiological roles. The learning resources for this video include the Chatterjee textbook of biochemistry, the biochemistry textbook by Mushtaq Ahmed, and your lecture notes. To begin, an acid is a substance, ion, molecule, or particle that yields hydrogen ions or protons in a solution. A base is anything that combines with hydrogen ions, acting as a proton acceptor. Acids and bases are categorized based on their dissociation. Strong acids completely dissociate, even in strongly acidic solutions, while weak acids only partially dissociate. Similarly, strong bases completely dissociate at high pH, and weak bases only partially dissociate. A conjugate base is essentially an acid that has lost its hydrogen ion. Its formula is the acid formula minus the hydrogen ion. For example, the conjugate base of hydrochloric acid, HCl, is chloride, or Cl-. The concentration of protons in an aqueous solution is expressed as pH, which is the negative log of the hydrogen ion concentration. The formula is pH equals minus log of the hydrogen ion concentration. A pH greater than 7 indicates an alkaline medium, which can lead to alkalosis. A pH of exactly 7 is neutral. A pH less than 7 indicates an acidic medium, which can lead to acidosis. The more alkaline a solution, the higher the pH. The more acidic a solution, the lower the pH. Water is neutral with a pH of 7. The pH of arterial blood is normally maintained between 7.35 and 7.45. The dissociation constant, Ka, indicates the acid strength. A larger Ka means a stronger acid because more of the acid has dissociated. Conversely, a smaller Ka means a weaker acid. The Henderson-Hasselbalch equation provides a quantitative relationship to calculate the pH of a solution based on the pKa of the weak acid and the ratio of conjugate base to weak acid concentrations. The Henderson-Hasselbalch equation has several applications. You can calculate pKa from pH and the concentration of the proton acceptor and donor. You can also calculate pH from pKa and the concentration of the proton acceptor and donor, or determine the ratio of proton donor and acceptor based on pH and pKa. Various buffer systems maintain the human body's pH balance. These include the carbonic acid, bicarbonate, protein, phosphate, ammonia, and hemoglobin buffer systems, each with differing capacity. Buffers play a critical role in controlling body pH by accepting or donating protons in response to changes in hydrogen ion concentration. These systems, like the bicarbonate buffer, operate optimally within specific physiological ranges. Enzymes also require specific pH ranges to function effectively. The respiratory and renal systems work alongside buffers to regulate pH. Now let's think like a clinician. If a 70 kilograms male suddenly loses 2 liters of blood, which fluid compartment is affected first? And will the intracellular fluid volume change immediately? Here is a riddle to consider. I move freely but carry no passport. I shrink cells and swell them too. Sodium commands me silently. Who am I? We begin with the kingdom of fluidia, centered by the total body water, or TBW, making up about 60% of an adult male's body weight. Body fluids transport nutrients, remove waste, regulate temperature, and maintain pH balance. The two provinces or types of body fluids are intracellular fluid, ICF, and extracellular fluid, ECF. ICF is the largest, about 2/3 of total body water, found inside cells and rich in potassium and magnesium. ECF is 1/3 of total body water, surrounding cells and including interstitial fluid and plasma. Total body fluids average 42 liters, which is 60% of body weight. Intracellular fluids make up 28 liters, or 40%, and extracellular fluids account for 14 liters, or 20%. The intracellular fluid compartment, 28 liters or 40%, consists of cytosol, which is 90% of ICF, and organelles, which is 10% of ICF. The extracellular fluid compartment, 14 liters or 20%, is composed of interstitial fluid, which is 11 liters or 3/4 of the ECF, and plasma, which is 3 liters or 1/4 of the ECF. Interstitial fluid facilitates substance exchange between blood and cells. Plasma transports nutrients, gases, waste, and hormones, and maintains osmotic pressure. Both contain electrolytes and other solutes, with plasma also rich in proteins. This diagram illustrates the distribution of fluids throughout the body, showing the relationship between intracellular and extracellular compartments and the components within the ECF. Next, let's introduce the physical properties of fluids. 3 important properties we will discuss are surface tension, viscosity, and osmosis, which are essential for understanding fluid behavior and functions. Surface tension is the force causing liquid molecules at the surface to pull inward due to cohesive forces. Water exhibits high surface tension because of hydrogen bonding. This inward cohesive force, or surface tension, leads to the formation of a, quote, surface film, allowing liquids to form droplets and exhibit capillary action. Surface tension is influenced by temperature and the nature of the liquid. In lipid digestion, large lipid globules have a small surface area, which hinders enzyme action. Bile salts reduce the surface tension, allowing lipids to form smaller, stable emulsion droplets. This increases the surface area for efficient enzymatic breakdown into glycerol and fatty acids. This image further visualizes how bile salts help to emulsify lipids by reducing the surface tension of large lipid globules, making them accessible for digestive enzymes. Viscosity refers to the resistance of a fluid to flow and is a measure of its internal friction. Think of how thick or thin a fluid is. High viscosity fluids like honey flow slowly, while low viscosity fluids like water flow easily. Viscosity is crucial as it affects processes such as blood flow, digestion, and joint lubrication. Resistance to blood flow is directly proportional to both the length of the blood vessel and the viscosity of the blood. Higher viscosity or longer vessels increase the resistance. Next, let's discuss osmosis, a term from Greek meaning push. Osmosis is the movement of a solvent, typically water, through a semi-permeable membrane. Thank you for your attention throughout this video on body fluids and their critical physical properties.